Okay, hello everyone, welcome back. So this is week 7 of the Experimental Biochemistry course. This course is coordinated by Professor Dasgupta from the Department of Chemistry and Professor Dev from the Department of Bioscience and Biotechnology, IIT Kharagpur. And I am Bias Mukherjee, a research scholar from the Department of Bioscience and Biotechnology. And today we are going to discuss about isolation and purification of proteins. Okay, so we have talked about different chromatographic techniques for analysis of proteins, for separation of proteins, right? We have talked about SDS page, native page, etc. in our previous class to study the different proteins in a mixture. Today we are going to talk about the entire process of making protein in the lab and purifying it. We will also talk about extraction of proteins from natural sources, and we will also talk about the disadvantages in that case. And that is why people came up with making recombinant proteins in the lab, so that we can get a lot more protein and a lot purer protein through a faster and more controllable strategy. Okay, so protein purification is a very simple topic, but it is very diverse. There are a lot of players in this full process, and we are going to talk about all of them today. Okay, so let's begin. Right. So isolation and purification of proteins. Now, why is isolation and purification of proteins required? That is because it has applications in all of these different disciplines that you can see. Right. So isolation and purification of proteins involves separating a specific desired proteins from other cellular components to obtain it in a pure and concentrated form. So high quality pure protein is crucial for accuracy and reliability of studying protein structure, function, and for applications in biotechnology, medicine, drug design, vaccine development, and other proteomic studies. Okay, we can see that there are a lot of different fields in which pure proteins are necessary. Okay. For example, enzyme catalysis studies, and then structural biology studies, vaccine development, drug development, biopharmaceutical production, functional assays, proteomics, right, biomedical research and biotechnology, clinical diagnostics, etc. Okay, so now extraction and purification of proteins from natural sources. Okay, we will look into this now. So isolation and purification of proteins can be performed directly from natural biological sources such as tissues, cells, blood, or microorganism. Right? The process typically involves cell disruption, homogenization, and sonication. So homogenization means that all the components are mixed together and they are homogenized, and in every small portion of that particular liquid you will find exact same composition as in another portion of that particular liquid, right? So that is called homogenization. And then centrifugation is done for separation. Precipitation is often done using different salts, for example ammonium sulfate, which is used for salting out of proteins. We will talk about this in the upcoming slides. Okay, and chromatographic techniques such as ion exchange, size explosion chromatography, and affinity chromatography. So in our last class, we discussed extensively about different chromatographic techniques. And in today's class, we are going to talk about only a few chromatographic techniques in very little details. Hello and welcome back. So in today's class, we are going to talk about isolation and purification of proteins. Okay, it would be great if you can answer in the class. Okay, so we are going to discuss a lot of MCQ questions, a lot of numerical questions, and a lot of multiple select questions. Okay, right. So okay, people are not interested in this topic, it seems. Anyway, so we'll carry on. Now, the main advantage is that the proteins are obtained in their native form with natural post-translational modifications. However, this approach can suffer from low yield, complex purification steps, and variability between biological samples, making large-scale production very challenging. Right? So, the problem with extraction from natural sources is that it is very low quantity is obtained in the pure form, and that is why reproducibility becomes a major issue, because oftentimes when we are extracting from this natural source this particular, let's say, a protein, it may be interacting with a lot of different other proteins. It can have different post-translational modifications. It can be obtained in the form of some different isoform as compared to what we were wanting. That is why the reproducibility of the experiments becomes a major challenge. Okay. Now, disadvantages are low yield, complex purification steps, and variability between samples. Advantage is that native form with natural post-translational modifications can be obtained. So what is the procedure? You can see that from the natural sources, tissue, cells, blood, and microorganisms, cell disruption is performed using homogenization and sonication, and then centrifugation is done, then precipitation using ammonium chloride is performed, and then different chromatographic techniques such as ion exchange, size exclusion chromatography, affinity chromatography, etc. is performed for the purification step. Okay, right. So now this particular figure is taken from a book chapter, okay, in which they are talking about extraction of proteins from natural sources in a lot of details. So I have provided the DOI, or digital object identifier, of that particular paper in the slide, okay, at the left bottom corner of the slide. So you can refer to this paper, and what we are seeing here, that is described in the next slide. So we will look at that quickly. So there are two different methods. One is wet extraction, another one is dry extraction. But before that, there are different processing steps for these grains or seeds. Okay, so what are they? They are dehulling. That means you're removing the hull, or the covering of the seed, defatting, soaking, drying, and milling. Milling means grinding, to produce a fine powder suitable for extraction. Then protein extraction can then proceed through wet extraction methods, where the proteins are released during cell disruption techniques using ultrasound, microwave treatment, high hydrostatic pressure, pulse electric fields, or enzymatic treatments, followed by protein extraction using conventional methods, aqueous or acid and alkaline methods, then salt and alcohol extraction procedures, right? Or novel approaches such as reverse micelle, and then subcritical water extraction and aqueous two-phase systems. So you can read about these if you're interested. Okay, so the extracted proteins are subsequently concentrated and purified using techniques like isoelectric precipitation, membrane processes such as ultrafiltration and dialysis, micellization, after which heat treatment and spray drying produces a final product, that is the isolated protein, and it is concentrated, right? So alternatively, a dry extraction method can be performed, such as sieving, gravity separation, air classification, and electrostatic separation can fractionate protein-rich particles directly from milled material without solvents. So you can see that this is a very extensive, very long procedure which can be followed for wet extraction methods. And that is why scientists came up with these alternative strategies. Okay, so we have two MCQs, and we can answer the two MCQs first, and then we can carry on with the other techniques. Okay, so for the first MCQ, the question is, during isolation of proteins from seeds, from plant seeds, using wet extraction, the primary purpose of isoelectric precipitation is to A. Increase protein solubility in aqueous buffers, B. Separate protein based on molecular size, C. Reduce protein stability by adjusting pH to the isoelectric point, and D is to denature proteins to remove bound lipids. Okay. For the second question, which of the following cell disruption technique enhances protein extraction by generating transient pores in the cell membranes using short bursts of high voltage, ultrasound treatment, high electrostatic pressure, pulse field treatment, and microwave heating? So we will look at the right answers. The right answers for the first question, right answer is C, that is reduce protein solubility by adjusting pH to the isoelectric point. So at this pH, what happens is the protein has net charge 0, because of which it precipitates, and there is this protein-protein interaction and formation of aggregates, and it ultimately precipitates and therefore helps in separation from the rest of the media. Okay. For the second question, the right answer is pulse electric field treatment. So in pulse electric field treatment, this applies short high voltage pulses that create temporary pores in the cell membrane, that is electroporation, the process, and increasing the permeability and improving the release of intracellular proteins. So these pores are created in the membrane of the protein, and then the proteins are released through these pores. Okay, so that is what is done. For the next strategy, it is overexpression and purification of recombinant proteins. Now, what are recombinant proteins? To not go into the trouble of isolating proteins from the natural source, people came up with this process of protein overexpression and purification in a desired host cell. Okay, and this protein, this engineered protein, is called recombinant protein. So what is this recombinant protein? It can be completely natural protein, but it is inserted into a plasmid. Okay, so the different steps we are going to discuss now. First is, there is our gene of interest. Now this can be a natural protein, it can be your mutated protein, it can be two different proteins, and it can be anything. Okay, it can be a chimera, it can be absolutely anything. So the gene that codes for that particular protein of interest, that is known as the gene of interest. So this gene of interest is taken and two restriction sites are added to the gene of interest. Now cloning can be done in different methods. Okay, so there are different ways of cloning. The most common and the most simplest one is using two restriction sites, and there are two restriction sites in the vector. So when we restrict and digest the vector, then what happens is, for example, this is our gene of interest with these two restriction enzyme sites, and this is the vector with two complementary strands of the same restriction enzyme site. It is going to come and join. So this is what ligation is, right? And this plasmid is formed, and then this is the expression plasmid. So that means it contains all the sequences that are needed for protein expression. And then what happens is, that is put into a host organism. The host organism, the cheapest one, and if you don't need any post-translational modification, then the host organism that you can use is bacteria. Okay, it grows the fastest, it is relatively cheaper than the other methods, and it is giving you a lot of protein at a time. So bacterial culture, it is grown and it grows overnight. So for protein expression, it is grown for a specific duration, that can be 5 hours, that can be whatever time your protein requires. And then what happens is, the bacteria is broken open by sonication. It is extracted, all the proteins, they are sonicated, centrifuged, and then the soluble part is removed and the soluble part is filtered, and then it is passed to the chromatography column. You can use affinity chromatography, and then after that you can go for size exclusion chromatography, or you can go for ion exchange chromatography, whatever suits you. And then you will go for protein structure and functional analysis after concentrating the protein. In between, you will also run an SDS page to check the purity of your protein, and also the concentration of the protein can be determined either using SDS page based techniques, or if the protein has aromatic residues, that is well and good, so you can go for absorption spectroscopic techniques. Okay, fluorescence spectroscopic techniques also. Now, so what we will do now is, what are the advantages of overexpression and purification of protein? The first one is definitely very high yield. The second one is customization. So in that gene sequence, you can, I told you that you can make any modifications that you want, right? So the proteins will have your specific modified sequence then. Okay, and it can have tags, it can have this histidine tag for affinity based chromatography. You can add one protein in the beginning, another protein at the end, so then two proteins will be formed together. All of these different things can be done. It is cost effective, it is cheaper, it is reproducible, because so keeping the conditions same, the bacteria is going to give the same protein every time. And then purity, of course, it is going to be very, very pure. And speed and efficiency, it takes very less time compared to other types of protein production strategies. Okay, right. So now we will look at the steps in a little more details. So if you see, gene cloning involves isolation and amplification of a desired gene. The gene is then inserted into a plasmid, which is a small piece of circular DNA that can be easily replicated and transferred into a host organism. Right. So something called codon optimization need to be done. So what is codon optimization? For example, in the body of that bacteria, let's say that for one particular amino acid, let's say for tryptophan. So for tryptophan, there is only one codon in bacteria, and that is UGG. But for, let's say, leucine, there are many codons. For other amino acids, okay, there can be many codons. So now amongst these many codons, let's say one codon is 40% of all the codons is just that codon. Okay, and then let's say 30% of all the other codons for that particular amino acid is the second codon. And then remaining, let's say your 5% is another codon, remaining let's say 10% is another codon, right? So this is called codon bias. So based on the availability of the tRNA for that specific codon, we can say whether one codon is biased, it is preferred more in a particular bacteria for a particular amino acid, right? So that is how, based depending upon the number of tRNAs present with the specific anticodon loop corresponding to that specific codon, it will be the gene sequence has to be optimized, right? So we will have to choose the codon that is maximum, that is most abundant in that bacteria. Sometimes what happens is we choose the second best codon because we want to, let's say, do some kind of modifications or something, and the best one is maybe taken for something else, right? So anyway, that is what we need to do. So next what happens is the host organism is then transformed with the plasmid using techniques such as electroporation or chemical transformation. This allows the host organism to express the protein encoded by the inserted gene. So expression of the protein involves inducing the host organism to produce the protein. This can be done by adding specific growth conditions or by adding a chemical inducer. The protein is then harvested from the host organism, usually through centrifugation or filtration. So what is induction? Induction means that you are telling the host organism to start producing the protein now. Until then, it will not start producing that particular protein. Now, why is that so? Because let's say a particular organism, it is supposed to produce the proteins that are needed for its own body. But then if it is unnecessarily producing huge quantities of a particular protein that is not even required by its own body, then it becomes very, very toxic for that organism. And so what happens is the organism eventually dies. So here what happens, the amount of protein that we could have got if the organism would have divided and formed a lot more organisms by then, that is not happening. So we are getting much less protein if the organism dies early. That is why we have trained the system in this way. We have modified the system in such a way is that that organism is having a genetic makeup where we can tell it to start production of our protein of interest. Until then, it will just normally multiply as it does. Okay. Now, expression of the protein involves, okay, inducing the host. That is done. So, the purification of the proteins is a vital step in the process. It involves removing any unwanted contaminants and impurities, which can be accomplished through various techniques such as chromatography, precipitation, and filtration. Okay, now the entire process of protein purification that is discussed here. Okay, so this is the most important slide. So let's look at it. So expression in E. coli. So in this huge bacterial culture, there is a lot of Fluidia berthini broth, and LB is like ready-made food for bacteria. So LB is taken and you only need to add a lot of water and then autoclave it, and so LB becomes this very, very nutrient-rich broth for bacteria, right? And then the bacteria is first taken in a primary culture in a small test tube, and then it is grown overnight and it reaches the OD of around 2 or so. And then what happens? let's say 1% or 5% culture is taken and it is added into the big LB flask. So how much is 1% culture? Let's say that if there is 1 liter of culture in this big flask, then 10 ml of LB will be taken and it will be put into this big flask, right? 10 ml of LB containing bacteria with OD2 that had grown during the primary culture. Anyway, so then this one grows for a particular amount of time at 37 degrees centigrade until it reaches an OD of 0.6. So what is that? That means the cell count has reached at a specific value such that it is ready for induction. Now if I induce, I will get enough proteins. Right? So at 600 nanometer, the wavelength is checked and that is 600 nanometer wavelength, the absorbance is checked and that is called OD 0.6. Hello and welcome back. So in today's class we are talking about protein isolation and purification. Okay, so we have a lot of questions in today's class also. It would be great if you can answer in the chat box. Okay, so right now we are talking about harvesting the E. coli cells. Okay, so we have taken this huge flask containing LB in which we have introduced the bacteria and the bacteria is growing at 37 degrees centigrade for let's say around 2 to 3 hours. So within 2 to 3 hours it will reach an OD of around 0.6. So that means at 600 nanometer it is giving a specific absorbance and that is sufficient for us to induce it now. So what is induction? Induction means that we are adding a specific chemical compound which is called IPTG in this case, and that is telling the bacteria to start making the protein. Okay, and then we will grow it for let's say another 5 hours. Okay, then after that we will spin it down. Okay, and we see that all the bacterial pellet, all the bacterial cells, they will come as and form the pellet at the bottom. All the media that will be on the top. So we will throw the media, we will take the bacterial pellet, we will dissolve it in a wash buffer. Okay, it is a tris buffer and we will dissolve it in that tris buffer and you can store this pellet for months. So we will add PMSF and PI cocktail and we will add sodium azide. So what are all of these different things? These are telling the bacteria that no fungal attack will be there on the bacteria. Second thing is the proteins wouldn't be broken down by the proteolytic enzymes. Okay, sodium azide is preventing fungal attack on the bacteria and PI cocktail and PMSF, these are preventing any proteolytic enzymes to break down proteins. So these wouldn't be needed right now, but during the sonication step when the proteins come out into the media, these would be required. Okay, and then what we will do is, if you look at the figure, then we will go for addition of lysozyme enzyme. Okay, and lysozyme is added and then it is incubated for a particular amount of time, then it is sonicated and then all the bacteria are broken open because of the presence of lysozyme plus sonication, and all the proteins that were inside the bacteria, they come out. We are adding DNAs and RNAs to remove the DNA and RNA present in that protein. If it is binding to that protein very tightly, then it is very important that you add DNAs and RNases, okay, if the protein has binding affinity for nucleic acids. Anyway, so next we will spin it again and all the protein, if it is soluble, it will go into the supernatant, but if it is not soluble, it will come into the inclusion body and all the bacterial cell debris, all the broken pieces of the bacteria will come and form the pellet at the bottom. Then what we will do is we will take this soluble lysate that you can see in the figure and we will pass it through a column. So normally we go for different chromatographic techniques such as anion exchange chromatography, cation exchange chromatography, affinity chromatography, okay, whatever you want to do, you will go for it. And then in the flow through, all the other bacterial proteins will go. In the column, the bacterial protein of interest will remain stuck. Then you will add this buffer with high amount of imidazole that you are looking at in this figure. Then the protein of interest is eluted and it is collected. Then this protein of interest, you can go for another round of purification using size exclusion chromatography to separate out any other different bound proteins or broken fragments of the proteins of your interest if there are any, and then we will change the buffer. So for that also size exclusion chromatography can be done. Otherwise dialysis can be done where buffer exchange is happening and the protein is kept in the buffer. That is normal for its physiological activity. Okay, for its activity. Then after that you can see that they have gone for the size exclusion chromatography and then run an SDS page. So in SDS page you can see there is one single clear band that tells us that the protein is pure and it is having a lot of high concentration, right? So that is the entire process of protein purification from bacteria. Now we will look at each of these steps of protein purification in details. Okay, so we'll start with the vector. So the vector in which you're inserting your gene of interest. Now vectors are of two different types. One is cloning vector, the other one is expression vector. So what is the difference between the two of them? So in cloning vector, this is used to acquire multiple copies of the foreign DNA fragment or the gene of interest, and they have features like the ORI, that is origin of replication, restriction sites, antibody resistance, and reporter gene. But for expression vector, there are a lot of other components. So what are these? An expression vector is utilized to acquire the gene product, which might be an RNA or a protein or the DNA of interest. They have similar features to a typical vector and also have regulatory elements like promoter, enhancer, termination sequence, and transcription start site, translation start site, transcription start site, then initiation site and stop codon. So all of these different components need to be there to ensure that this gene of interest is transcribed into mRNA of interest, and then the machinery of the bacteria forms proteins from there, right? So all of these different sequences need to be there in the expression vector. Now, what kind of a host do you need? Now, you have taken the vector, you have put your gene of interest into the vector. Now, you want to insert the vector into an organism so that there it multiplies and produces your protein of interest. Now, how are you going to decide what organism you want as your expression system, right? So, that depends upon the type of protein that you need to produce. So, if your protein has no post-translational modification, if its size is relatively small, then bacteria would be your best choice. So you can see that bacteria is low cost, has rapid expression, easy to scale up, so you can produce in large quantities, no post-translation modification is required, but uh, it will the protein can go into inclusion bodies if it is not very soluble, and then there is a limitation with molecular weight. So two very large molecular weight proteins cannot be made in bacteria. Okay. Then there can be mammalian cells, for example, HEC293 and CHO cells. So here what happens is very good post-translational modification machinery is there in these human cells, and that is why soluble proteins can be prepared using these. Okay, but then the disadvantage is very long culture time, plus this is also very expensive to maintain. Okay, then the other one is insect cells. Now insect cells are also useful if you want some specific post-translational modifications. They are also relatively cheaper compared to mammalian cells, and they form these soluble proteins. Okay, but it is relatively costly compared to bacterial culture. Then comes yeast. So yeast is only useful for very specific post-translational modification. It is relatively cheaper than insect cells and it has rapid expression. Okay. So these are the different pros and cons of using different uh, expression systems. Okay, here a big chart is given for you and all the challenges, the advantages and the most common applications are given. So you can go through this whenever you get time. Okay, so whatever we discussed in the previous slide is only given in much more details. Now here, this is taken from one paper where a specific, the most commonly used bacterial cell that is used for protein purification, protein overexpression, that is explained here and its different variants are shown. Okay, so the cell is known as BL21. This is a variant of E. coli. So BL21 is E. coli polymerase, but no T7 RNA polymerase. So this is the natural form. Okay, Okay, so this is used for expression of non-toxic proteins, and it can be used for expression of plasmids such as PGEX and PMAL. Then there is BL21 DE3. Now what is this DE3? This is actually taken from a bacteriophage or a virus. So the genetic material of this particular bacteria BL21 has a portion of the genetic material taken from a virus incorporated into it. That is from lambda DE3 phage. Okay, so the lambda DE3 phage region containing T7 RNA polymerase, T7 phage RNA polymerase, from that lambda phage, that gene is taken and integrated into this bacteria. So this bacteria can now produce this viral RNA polymerase. Why? Because viral RNA polymerase is 5 to 10 times faster and 20 times less error prone compared to the bacterial RNA polymerase. Right? So what happens is a lot more protein gets overexpressed in a very less time. And BL21 allows the simultaneous expression of both T7 RNA polymerase and E. coli RNA polymerase. So this is suitable for expression of toxic proteins and non-toxic proteins both. Then, this is useful for efficient purification of genes cloned in the expression vectors containing the T7 promoters such as PET series. So this is what we also use in the lab. Okay, and for the protein expression of the plasmids such as PGEX and PMAL. Next we have BL21 DE3 P-lyse S. So this is the same system but with another plasmid. So this particular plasmid is P-lyse S. So it produces a specific kind of lysozyme which cleaves off the T7 RNA polymerase that is formed before induction. That is, we look at this and then you'll understand. So this carries the P-lyse S plasmid having chloramphenicol resistance and containing gene expressing T7 lysozyme. So this is a protein that cleaves off, one second. Yeah. So this is a protein that cleaves off T7 RNA polymerase when it is produced in very small quantities and it is produced before induction. Okay, so we will discuss this in details very soon. Okay. Now, this is suitable for expression of toxic proteins and non-toxic proteins both. Right? So T7 lysozyme can reduce the background level expression of the target gene, but it does not interfere with the IPTG induced expression. So it is basically helping you to control the background expression of this T7 RNA polymerase when it has not been induced by us, right? Now we will talk about this BL21D3 P-lyse S in a little more details. So if you look at this particular bacteria, you will see that it is having this E. coli genome. In the E. coli genome, DE3 phage genetic material is encoded. And this phage genetic material actually codes for T7 RNA polymerase. So it is having the gene for T7 RNA polymerase. And that gene, at the promoter region of that gene is E. coli RNA polymerase. So when E. coli RNA polymerase comes and binds to it, this will get activated. Also there is this LAC promoter. So you know that LAC promoter is under the repression system by LAC repressor protein. And this has been modified in such a way that this LAC promoter is actually converted into LAC UV5 promoter, and this LAC UV5 promoter is a lot more potent compared to just normal regular LAC promoter. Now if you look at this figure, let's try to understand what happens. Normally this LAC promoter, it is a leaky promoter, so there will be some leaky expression. So that means some amount of T7 RNA polymerase will get produced even when no IPTG has been induced, right? So this small amount of T7 RNA polymerase that gets produced, this can go and inside our PET vector, this vector is inserted into the bacteria. So in the PET vector there is the gene of interest, and this gene of interest is under the control of the T7 RNA polymerase promoter. So this T7RNA polymerase will go and bind to the promoter region and start producing a gene of interest. But that is toxic for the bacteria because this can be a toxic protein and it will kill the bacteria. To prevent that, there is this other plasmid which is P lyse S or E, and it produces this proteolytic uh, enzyme or protein, and it goes and binds to this T7RNA polymerase produced in small quantity and inactivates it. But then when you are adding IPTG, IPTG is an analog of allolactose. Okay, so when you are adding IPTG, it will go and bind to the this lac repressor and prevent the lac repressor from binding to the E. coli RNA polymerase promoter region. That is why E. coli RNA polymerase will be sufficiently able to sit and bind to this promoter and synthesize T7 RNA polymerase in huge quantities. When it is synthesized in huge quantity, P lyase S is now, it cannot bind to all of those molecules. So now a lot of T7 RNA polymerase is getting produced. It is going and binding to our gene of interest promoter, and our gene of interest is getting expressed. So we are getting a protein of interest. Right. So this is the entire mechanism of function of the BL21DE3 protein expression system. Okay. Now we have a question. You can try to answer this question in the chat box. Okay. So a researcher wants to express a human glycoprotein enzyme. So remember, glycoprotein enzyme, that means there is some kind of modification that requires a proper disulfide bond formation and N-linked glycosylation for activity. Now, which of the following expression system would be most suitable for producing a functional recombinant protein? One is E. coli BL21 D3. Saccharomyces cerevisiae, which is yeast. Then there is baculovirus or insect cell system. And then there is mammalian expression system, for example, HEC or CHO cells. Right? So you need two things. One is a disulfide bond, the other one is N-linked glycosylation. 3. Right. Good. Let's see the right answer. Yes, very good. So, yes, 3 is the right answer because when it comes to E. coli, it cannot perform glycosylation. So, that is not at all possible. Coming to yeast, glycosylation is produced, but non-human hyperglycosylation is often done. So, that is also not appropriate for a protein. For insect cells, it performs eukaryotic post-translation modifications correctly, and for mammalian cell it would be the best choice. Okay, now we go to the other question, the next question. Okay, so the E. coli strain BL21DE3 is most commonly used recombinant protein expression using T7 promoter based vectors, for example PET system. Which of the following features contribute to its sustainability? Right, to its suitability. So A is presence of T7 RNA polymerase gene under LAC-UV5 promoter control. Deficiency of LON and OMP-T proteases reducing the recombinant protein degradation. Ability to perform glycosylation modifications. IPTG induction leading to expression of T7 RNA polymerase, which drives the transcription from T7 RNA, T7 promoter. Okay. So, which all do you think would be correct? 2. Yeah, very good. That is correct. 2 is correct. That is because this LAC-UV5 promoter, this is a modified form of normal LAC promoter. How? Because it reduces the dependence of this LAC promoter system to get activated based on CAP and cyclic AMP activation. Plus, it drives very strong and more reliable transcription. Another thing is that BL21D3 really lacks these two proteins. One is LON and the other one is OMP-T proteins. These proteins would ensure, lacking these two proteins would ensure that there is no degradation of the proteins that are produced by it. Okay. And IPTG induction, you guys know, and it cannot perform complex glycosylation, so only that is incorrect. The others are all correct. Okay. Now we have another question. A recombinant protein expressed in E. coli BL21D3 forms insoluble inclusion bodies. Okay, insoluble inclusion bodies. Which of the following strategies are likely to increase soluble protein expression? So this is a very practical problem in a protein biology lab. If the protein is rendered insoluble, then what can you do? So one of the ways is lowering induction temperature from 37 to 16 degrees centigrade. So to prevent the protein misfolding. Okay, you're giving more time and less temperature to the bacteria to fold its protein. So it is grown for a longer duration, let's say overnight. That is one way. The other one is reducing IPTG concentration. So when you reduce IPTG concentration, less amount of protein is produced at a time. Increasing expression rate using stronger promoters and fusion with highly soluble tags such as MBP or GST. Okay. Achha. So you are saying that it is A, B and D. Good. Let's see the right answer. Yeah. So that is very correct. A, B and D would be the right choices to make. Right. So lower temperature means slower translation, better folding. Lower IPTG, reduced expression rate and less aggregation. Stronger promoter means faster expression and more propensity to misfold. Correct. Solubility tags are often very helpful in promoting folding. Okay. Next we will go for these the process of entire recombinant protein production. Okay, so we will start with gene cloning. So insertion of the gene of interest into a suitable plasmid vector under the control of a strong promoter. That is number one thing that you need to do. Second thing is transformation. So transferring the recombinant plasmid into the competent bacterial cells using the process called transformation. So entering this vector into the bacterial cell. Cultivation and growth. So growth of the transformed bacterial culture in a suitable growth media, often LB or Ludia Bertini broth. Induce protein expression by adding an inducer. This inducer is normally isopropyl beta D1 thiogalactopyranoside or IPTG. So what is this IPTG? IPTG is actually a molecular mimic of allolactose. So non-metabolizable. So that means if you are adding normal allolactose, then the bacteria can eat it up. It will metabolize it and it will be over. And then the bacteria will stop making the protein. But we don't want that. So we want the bacteria to constantly think that there is allolactose in the medium. I should activate my lacoperon system, but then I'm not able to digest it. I'm not able to use it up, and so it will consistently start making the protein. So this triggers transcription of the lacoperon, and it is used to induce protein expression when the gene is under the control of the lacoperon. So which gene is under the control of the lacoperon? T7 RNA polymerase production. That is under the control of the lacoperon. Okay. Cell harvesting. Harvest the bacterial cells after a specific incubation period of often 5 hours or more. That is 7 degrees centigrade, 5 hours, 16 degrees centigrade overnight. Okay. When the protein of interest is expressed at high level. Now resuspend the pellet containing bacterial cells in lysis or wash buffer with a protease inhibitor such as PMSF and sodium azide to prevent fungal growth. So PMSF, the structure is given there for your reference. Now, steps in protein purification, cell disruption. So, disrupt the bacterial cells to release the intracellular contents, including the recombinant protein after adding lysozyme. Lysozyme is the one that breaks the cell wall of the bacteria by sonication. Okay, centrifugation, you're spinning the cells and all the insoluble cell debris come to the bottom in form of pellet. All the media, all the soluble portion of the proteins, the DNA, RNA, everything, they go into the soluble portion and the supernatant is collected. Okay, and purification. So employ chromatographic techniques for purification. dialysis and concentration is done. Again, instead of dialysis, you can exchange the buffer using size exclusion chromatography, and that will add another layer of purification anyway. So dialyze the protein into a suitable buffer and concentrate it to get the desired concentration. Quality check is very important. So SDS page is run for quality check and confirm the purity and molecular weight of the protein. Storage. Store the purified protein under appropriate conditions to maintain the stability by adding PMSF and PI cocktail. Always remember, protein needs to be stored safely with the addition of PMSF and PI cocktail, otherwise it will be cleaved by the proteases. Okay. Structural and functional assay is then performed for the proteins. Now, these are the different techniques of protein separation. So, precipitation of the protein is done by using isoelectric focusing of the protein, salting out of the protein, right? And then centrifugation is done. So, there can be density gradient centrifugation, there can be differential centrifugation, membrane separation, so ultrafiltration and dialysis can be done. So all the separation based on different properties that is mentioned, and resolving power is also mentioned. Chromatography has very high resolving power. So there is gel filtration chromatography, ion exchange chromatography, hydrophobic interaction chromatography, and affinity chromatography. Okay, so all of these exploit different properties of the protein for their separation. So we'll talk about salting out. Now, interestingly, when you're adding very little amount of salt in the protein, then what is happening? The at lower concentration, this is called salting in. So what happens is this salt is allowing the protein to interact more with water and be more soluble. Okay, so at lower concentration, added salt usually increases the solubility of the charged macromolecule because the salt screens out charge interactions. So low salt prevents aggregation and therefore prevents precipitation of the protein. But when a lot of salt is added, as you can see the curve, solubility curve, what happens is all these water molecules, they start interacting with the salt molecules. Then what happens, the protein molecules, they are left alone, they all interact with themselves, and then all of them come together, they will interact with themselves and they will form aggregates and it will get precipitated out. Okay. So high salt removes the solvation sphere, that is around the protein there is this ordered arrangement of water molecules, right? So that is called solvation sphere from the protein molecule and they come out of the solution. Now there is this arrangement of salts and this is called the Hofmeister series. So here a lot of different salt molecules, ions, they are all ranked based on their ability to stabilize or destabilize proteins in solution and influence the solubility. So sulfate is the most salting out, causing anion. Okay, and then as you go down the series, the salts in the right, they are called chaeotropes, and the salts in the left, they are called cosmotropes. So stabilize protein structure and promote salting out, and the chaeotropes, they destabilize the proteins and may increase solubility or cause denaturation. Okay, so these are the different strategies of salting in and salting out. Now, you have already read about ion exchange chromatography and affinity chromatography. So, in this slide, in this class, we have one slide for each to just give you a very brief understanding of all these two techniques once again. Right? So, if you remember, in anion exchanger, the matrix was positive, the protein was anionic, right? And then what happened is negatively charged anion or analyte binds to the anion exchanger which is positively charged, and then initially there is low salt, so all the flow through the positively charged protein is eluting at low salt, and then when you increase the salt, the coulombic interactions are reduced and the bound proteins or the negatively charged proteins, they are all eluting at high salt concentration. So negatively charged proteins and positively charged proteins and positively charged stationary phase. So all of these are shown in this diagram, right? In our last to last class, this entire strategy, entire technique is given in a lot more details on the pH, and the pH of the buffers is explained there, and the different resins are explained. Okay, so you can refer to that. Again, for NINTA, if you remember, we know that there was this protein of interest. The protein of interest has a tag of histidines. 6 histidines are there. 2 histidines at a time bind to one nickel, which is chelated by a particular compound called NTA, which is nitrilotriacetic acid, and this is bound to the agarose bead. Okay, so when the protein of interest are, all the proteins are flown through, then all the other proteins, they are flown through at first, they are coming into our flow through, and then when you are adding this buffer with very high amount of imidazole ring, which is the side chain of histidine, then what happens, these imidazole groups, they will go and bind to this nickel and our protein of interest will elute. Also, there is higher salt, there is both biospecific elution and there is high salt dilution, right? So FPLC exploits the NINTA affinity chromatography technique to purify his 6 stacked proteins. Elution is either biospecific with an analyte like competitor or non-specific increased pH or salt concentration. Okay, and this entire strategy is called immobilized metal affinity chromatography or IMAC. Right? So after purification of the protein, you can go for all of these different strategies. You can study the characterization. You can characterize the protein using mass spectrometry, which is needed to know the identity of the protein, whether the protein that you want is the right protein or not. Then there can be conformational stability you want to know, then functions of the protein, for example, biochemical, immunochemical, protein-protein interactions, protein-DNA interactions, protein-RNA interactions, protein-small molecule interactions, all of these different functions you want to know. And then also if you want to know the structure of the protein, then there is protein crystallization, X-ray analysis, NMR, electron microscopy, etc. Right? So all of these different studies are done for the different proteins. Okay, so structural study of proteins. Okay, right. So the crystals. So what has happened in so three different things can be done for structural study of proteins. One is X-ray crystallography and diffraction. The second one is CD spectroscopy, and the third one is NMR spectroscopy. Okay, so there are different steps for each, and we will just, I have the full explained procedure, strategy, advantages and disadvantages for each of these. You will go through them. Okay, but just for your understanding, I'll tell you about these in brief. So if you see, for XRD and for NMR, both of these can be used for studying the three-dimensional structure of a protein. But CD spectroscopy gives you the two-dimensional structure of a protein. Okay, the different process of XRD is the protein is taken and the protein is crystallized. Okay, specific salt concentration, specific protein concentration is maintained. For crystallization, this crystal is subjected to X radiations, and then what happens, the lattices in the crystal, they diffract the X-rays, and this diffraction pattern is taken on a map. This is called electron density map. Okay? So every electron, so basically every atom is diffracting the X-rays in different directions, and that is giving you this image of the electron density map. Then this map is studied, and then the amplitude is received, but then the phases of the waves are not received. So something called as phase correction is done. So that is a lot of extensive, very intensive work. And then the electron density map is used to construct, it is refined, and to get the atomic model. Reference structure is often used, and then finally that model is refined again and again, and the final crystal structure is obtained. Okay, for CD spectroscopy, we will read about this, right? So you can see that X-ray crystallography gives information about the atomic 3D structure, and it is very useful for very large proteins, huge proteins. Okay, and the limitation is that not all proteins form crystals. Okay, plus the solving the structure is also very, very intensive work. Now X-ray crystallography, so you can read about this. So whatever I explained to you, that is only here. Okay, and CD spectroscopy, so this tells you about the secondary structure of the protein. It is a very fast technique, very little amount of protein is required, very simple strategy, but it is low resolution. So you can only know about whether a protein is forming more of an alpha helix or less of an disordered structure, but it is not going to tell you exactly where in the protein that change is happening, right? Then there is NMR spectroscopy. So solution NMR spectroscopy is very powerful. It tells you about the dynamics of the protein, okay, different regions of the protein, what is the dynamics, how it is changing, plus whether that particular protein is interacting with a particular ligand, with a particular DNA, protein, RNA, small molecule, etc. So you just mix both of them and then you study the peaks. Now what are these peaks? We will look at them, right? So here both CT spectroscopy and NMR spectroscopy is given in a lot of details. So you can read both of these, and if you have any doubts we can discuss in the next class. Okay, so I'll just tell you a little about these two techniques. So in NMR spectroscopy, a very well purified protein sample is taken, and if it is unlabeled, so there is just normal hydrogen, then also you can get a 1D NMR because 1H is also NMR active. And if you go for 15N and 1H labeled sample, so that means in normal N13 it is just normal, but then for N15 you will get resonance at characteristic frequencies. So each atom in a folded protein experiences a slightly different chemical environment due to the protein's three-dimensional structure. So their resonance frequencies will shift slightly. So even when you are applying only one single frequency to the whole protein, different regions in the protein will have different chemical shifts, or they will have different frequencies, right? And that is because of, let's say this atom is here, it is experiencing different chemical environment compared to another atom which is at a different place in the protein. Because of that, you will have different chemical shifts, and then the chemical shifts obtained in the proton axis by the nitrogen axis, they are plotted in two different axes, and then you will get these peaks like small mountains which are cut, and then you get these small round structures. Okay, and this solution NMR spectroscopy can help to reveal protein folding, conformational changes, interactions with other proteins or nucleic acids. NMR provides very valuable information about the protein dynamics and molecular interactions. Okay, now we will start with the question answers. Okay, now structural studies of proteins, there are all of these different strategies, different biophysical techniques, biochemical techniques for the study of different proteins. Okay, so in our course we will talk about a few of them as we proceed with the different classes, different lectures in different weeks. We will talk about different strategies. Okay, and this is taken from, I think, a review paper or a book chapter, so you can go back to that. I think the link is provided at the reference slide, so you can go back to that and you can read about this. Okay, so all of these different biophysical techniques are given in one place. So for your exams, remember, biophysical techniques are the places, these are the hot spots for questions. A lot of questions will be asked from here. Okay, so master all of these different techniques, different strategies very well. Okay, so we'll start with the question answers quickly. So which of the following salts are commonly used for salting out of the protein? Okay, one is ammonium chloride, one is sodium chloride, ammonium sulfate, and magnesium chloride. Remember the Hofmeister series and answer. Right, now for the next one. In a mixture of 4 proteins listed below, which should elute third in the size exclusion or gel filtration chromatography? Okay, simple, you should be able to do. Remember, at first the biggest one comes and at last the smallest one. Achha, C and B. Yes, let's see. Let's see the right answer. Yeah, very good. It is, sorry, this definitely should be C. So it is ammonium sulfate. Ammonium sulfate is the right answer. Okay. Yeah, so it is C and B. Okay. Okay. Now we have, which of the following techniques can be used to separate a protein that binds strongly to its substrate? Okay, so cation exchange chromatography, anion exchange chromatography, and then we have affinity chromatography and gel filtration chromatography. For the second question, gel filtration chromatography separates proteins based on their dash. Okay, surface charge and C and B. Okay. So surface charge, shape and size, affinity to the chromatographic column and PI. Let's see. Yeah, so it is C and B. Very good. So if you look at the different properties of the proteins and the different techniques, a table is given for you. So diameter of the molecule, if we zoom in, you'll be able to see. So diameter of the molecule is size exclusion chromatography, electric charge of the molecule, ion exchange chromatography, presence of hydrophobic domain, reverse phase chromatography, presence of hydrophobic groups, hydrophobic interaction chromatography, and biospecific interaction is affinity chromatography. Okay. Now we go to the next question. So there are 3 questions in this slide. Very simple, straightforward questions. Try to answer. Right. So which of the following techniques can be used to study the secondary structure of protein? SDS page, CD, gel filtration chromatography, and dynamic light scattering. Then we have which of the following determine the size of a protein. Which of the following techniques can be used? Size exclusion chromatography, SDS page, analytical ultracentrifuge, and all of the above. So which of the following techniques can be used to determine the 3-dimensional structure of a protein? Dynamic light scattering, NMR spectroscopy, X-ray crystallography, and CD. BDB. Let's look at the right answer. Yeah. Okay. So B, D, B and C, all of these are right answers, right? So secondary structure, definitely CD. And for determining size of the protein, all of the above would be the right answer. And to get 3D structure of a protein, you have both NMR and crystallography. Okay. Right. So now we have 2 more questions. In order to lyse cells, sonication is used. This process breaks apart the cells by light energy, heat energy, sound energy, chemical energy. And then imidazole can bind to C and B. Okay. It is C and A because the first one is sound energy, that is correct. Imidazole can bind to nickel. It is the principle of NINTA chromatography. Right? Okay. So at 280 nanometer, a protein solution has an absorbance of 8 to 80 equals to 7 and the molar extinction coefficient of epsilon 280 of the protein is so and so. What is the concentration of the protein? Now do this calculation and I'll be back in a minute. Okay, and for the next one we have so for the next one we have protein absorbs 280 nanometer due to the presence of which amino acid? Okay. Protein absorbs at BDC. Okay, let's see what is the right answer. Yes, it is B, B and C. Okay, so let's see. Absorbance, you know the formula is A is equal to epsilon into C into L. So here A is given and that is 7. Epsilon is given and you know that L is 1 centimeter. So you have to calculate C and C is coming to be 0.52 millimolar. So B is the right answer. Okay. Now for the next question, at 280 all the aromatic amino acids absorbed. So if you have to mark only one as the right answer, it should be tryptophan because tyrosine is 275. Yeah, okay, you didn't see the unit. Okay, okay, no problem. Anyway, so tyrosine is 275 technically, and I think this phenylalanine is 265 or something like that. So to be very very technical, you should go for tryptophan. But if it is an MSQ, then maybe you can go for both tyrosine and tryptophan, right? So, but the safest choice, remember, would only be tryptophan. Okay, right. Now we have a tool. Now, since bioinformatics is also a part of this today's class, so let's say that you have a particular protein sequence. When you go to this website called Expeciprot and you put the protein sequence, it will give you everything about that protein. So that includes extinction coefficient, that includes the number of amino acids, molecular weight, hydrophobicity index, Gravy score, then theoretical PI, the number of charged amino acids, number of positively charged amino acids, number of negatively charged amino acids, everything. Right? So the entire full history geography of the protein that will be given. So now if you have this particular question, okay, so they have given, so this obviously wouldn't come for your exam, but it was a part of today's class, that is why we are discussing. So let's say that this particular protein sequence was given to you and they are asking you which of the following statements is true for this particular protein, right? So what you need to do is you just need to take the sequence and paste it in Expeci. So once you paste it, the website link is given for your reference. So you will come to know that the number of amino acid is 76, molecular weight is 9.5 kDa. So that you can see that is correct. A is correct. PI is 10.3. So PI 10, that is also correct. 10.3 PI, that is correct. Extinction coefficient is also given. Right, so all of these things are correct. So it will have maximum solubility at a buffer of 10. That is definitely wrong because that is very close to its PI. So it will be insoluble, right? So that is something that you can come to know about a protein just from the sequence of the protein. Okay, now this question is again important because the protein in the previous question is in a buffer of pH 7. It can be purified by which of the following chromatographic strategies? Okay. B. Okay. So you know that this is the PI and below the pH. Sorry. So if the pH is below the PI, then the protein is going to have positive charge. Correct. So that means it should be cation exchange chromatography. Yes. So it is cation exchange chromatography. And if this was an MSQ, then you could also answer size exclusion chromatography. Right. So since they are not talking about any binding partner or there is no his tag visible, then you cannot say that affinity chromatography can be performed, but then ACC normally can be performed. But if it is an MCQ, always only go for cation exchange. Okay. Now we have two questions which are gate level MCQ questions, and then that is the end of our chapter. Okay, that is the end of our class for today. Okay, let's try to answer these two questions. Okay, these are very practical, everyday life, experimental biochemistry questions. Now see, a recombinant protein expressed in E. coli accumulates primarily in the inclusion bodies. A researcher follows the purification strategy below. First, cell lysis, centrifugation to pellet the inclusion body. So all the protein comes in the pellet now, not the supernatant, remember. So washing the pellet with buffer containing Triton X100 and low concentration urea. So you are washing the pellet properly. Solubilization using 6 molar guanidine hydrochloride. Right, so all the other proteins and everything, they are getting washed away. Now you are solubilizing the whole pellet using guanidine chloride, and gradual removal of the denaturant by dialysis. Okay, so using dialysis, all the guanidine chloride is removed and you get the pure protein. So let's see which of the following statements correctly explain the purpose of steps 3 and 4. Okay, so step 3 removes the membrane fragments, loosely associated contaminants from the inclusion bodies. Step 3 solubilizes aggregated recombinant protein completely. Step 4 denatures the protein and disrupts non-covalent interaction to solubilize the inclusion bodies. Step 4 selectively cleaves disulfide linkage in the recombinant proteins. So which is correct? Which do you think is correct? Okay. Acha. Okay, no problem. We'll look at the right answer. So the right answer is A and C. Why? Yeah, sure. You could have guessed, no problem. Since we already saw the answer, we'll look at the answer now. Okay. So inclusion body washing, so often with detergents such as Triton X100 and mild detergent, removes lipids, membrane fragments, nucleic acids and loosely bound proteins without dissolving the aggregated recombinant protein. So that means A is correct. Now washing does not fully solubilize the inclusion body, it only cleans it. So it is cleaning all of these other components and removing them. So loosely bound, remember lipids, membrane fragments, nucleic acids and loosely bound proteins. All of these are getting removed. Okay, now 6 molar guanidine chloride that is added. It is a very strong chemotropic agent that disrupts hydrogen bonding and hydrophobic interactions, thereby solubilizing aggregated proteins. Right, so C is correct. Guanidine chloride does not specifically cleave disulfide bonds. Reducing agents such as beta-mercaptoethanol or DTT are needed. So that means option number 4 was incorrect. What about B? B is also incorrect because washing does not fully solubilize the protein, but then guanidine chloride was used for solubilizing the protein. So both A and C are correct and the other options are wrong. Okay. So these practical questions are very good for your concept. Okay, so these are very good conceptual questions. It's best if you practice them well. Now see, there is another question. This is the last question for today's class. So a recombinant protein expressed in E. coli forms insoluble aggregates. To improve the soluble expression, the gene is fused with maltose binding protein. Okay, this is a huge protein, helps with solubility. After expression, purification is performed using amylose affinity chromatography, followed by protease cleavage to remove MBP. Now, MBP was then attached to the protein, so you remove this MBP by using this proteolytic tag. Between MBP and the protein of interest, you put a tag and then you cleave. So it can be a thrombin cleavage tag, it can be any proteolytic enzyme cleavage tag. And MBP is removed, your protein of interest is taken, do an ACC, get your protein of interest. Okay, so which of the following statements correctly explain the advantages of using this MBP fusion? So this is called a fusion protein. Okay, now MBP enhances folding efficiency for many recombinant proteins, improving the solubility. Right, MBP allows purification using amylose affinity chromatography due to its specific maltose binding affinity. Its name is only maltose binding protein, correct? So if the column has maltose, it is definitely going to bind to it and it is going to assist in purification. Now, MBP prevents formation of inclusion bodies by covalently cross-linking target proteins. After purification, a fusion partner can be removed using a site-specific protease. So, Which one do you think is correct? Which statements correctly explain the advantages of using MVP fusion? Two. Two is A, B and D only. Okay, let's see. Yeah, so 2 is correct. So see, MVP is highly soluble. It acts as a solubility enhancer, often improving the proper folding of the fusion proteins. So A is definitely correct. Okay. MVP specifically binds to maltose or amylose resin, enabling affinity purification. Correct. MVP does not prevent aggregation through covalent cross-linking. It mainly acts through chaperone-like effect and increases solubility. So how does it help with solubility? It acts through chaperone-like effect. Right. Now, fusion constructs. So see, there are these protease cleavage sites. For example, TEV. There can be thrombin. There can be these other cleavage sites where you can use this protease to cleave off the solubility tag. Correct. So then A, B and D are the right answers. So that is it for today's class. Thank you so much for your attention. Thank you for your participation. These are all the references that you can go through. Okay, so there are 2 virtual lab references also. Protein purification by Affinity Chromatography and protein purification by ACC. So you can go through these. You know by now that there are these practical laboratory skills that you can learn by attending these virtual lab programs, right? Yeah, so I was also part of a group who created this particular, so we had these 10 different experiments, and all the links that I'm providing to you, you can see in the contributors list that there are these same names will get repeated. So there are 2 professors. One is my professor, who's Professor De, and he is the coordinator of this course. And another professor is Professor Shamunto. So he and my professor, both of them, along with, there was this simulation engineer whose name is Prakriti. And other than that, there were 4 of us. So there were 4 scholars. So all of us created. So individual of these experiments were given to us, and all the ones that are relevant to your course, I just keep on adding. Because this was done with a lot of care and a lot of information is there in that. So this would help you a lot to understand these concepts better. Plus you can visualize right in front of you through these different simulations. And so it would make it very interesting for you to understand the concepts. Okay. So that's all for today's class. Thank you. If there are no more questions, we can end the class. Okay, we'll see you again next week. Thank you. Yeah, let's end the call. Bye bye.